being organized in the context of the 75th anniversary of the independence of the Republic of India. The Ministry of Education, Tertiary Education, Science and Technology, and the High Commission of India have joined hands together to work to organize this India-Mauritius STEM Week 2022 with the collaboration of the Rajiv Gandhi Science Center. So today we are very pleased, we are very honored to have in our midst today Dr. Soumitra Das from the Indian Institute of Science to be the guest speaker in the field of genomics of SARS-CoV-2 vaccine. So before passing on to Dr. Das, I would like to invite Dr. Molu, the director of Rajiv Gandhi Science Center, for a welcome note.
Good afternoon, everyone who is joining us online today for this webinar, the third webinar in this series. Now, first of all, I would like to thank our parent ministry, the Ministry of Education, Tertiary Education, Science and Technology, under the patronage of the Vice Prime Minister, Honorable Minister Lila Devi Dukandachman, who is our Minister for Education, Tertiary Education, Science and Technology, who has been coordinating to ensure that this event get realized in the best condition. And of course, without the support of the High Commission of India, this project wouldn't have been possible. And it's the first of its kind because we call it the India-Mauritius STEM Week, STEM meaning science, technology, engineering, and mathematics, which we are at our center, we promote in various forms and ways. And one important thing this week is we have exhibitions, we have seminars live, public visits, and as well as a series of webinars. Now, as mentioned by Hemraj, we are privileged to have Dr. Das among us today from IISc Bangalore. Now, if you recall, every one of us, many people have taking or still take science for granted. However, when we had the pandemic, the COVID-19 pandemic, it was a shocker for the whole population. People were taken aback. And every step that government took was connected to science. Every step, wearing of masks, social distancing, etc., sanitizer, they're all connected to science, and we show the importance of science. And the major leap occurred when we started getting the vaccine. And I think this was a record time in which a vaccine was developed in the world. And to tell us more about, I'm very privileged and very thankful to the Indian High Commission to have been able to connect us with IISc Bangalore, especially Dr. Das, for this special webinar. So I will let you listen to him and we'll interact throughout the session. And I will pass the floor to Mr. Hemraj Ransaman to introduce Dr. Das and then let Dr. Das give his expose. Thank you very much, everyone.
Yes, thank you, Dr. Molu. In fact, we are very honored, very privileged to have in our midst today Dr. Soumitra Das, Professor and Sir J.C. Bose National Fellow from the Department of Microbiology and Cell Biology, Indian Institute of Science, India. Well, Dr. Das has a very rich career and has had a very difficult form. It's going to take me the whole time slot to read all his career achievements. Just I would like to mention a few. He has enjoyed a very rich career in chemical biology, in viral genomics research, and one thing which particularly attracted me is that he discovered a small RNA molecule from Saccharomyces cerevisiae, if I'm spelling it very correctly, and showed its selective antiviral activity against polio and hepatitis C virus. And his research work focuses on the biology and pathogenesis of several RNA viruses. Das has also been working on vaccine strategies. He has successfully coordinated the Pan-India 1000 SARS-CoV-2 RNA genome project of DBT, Government of India, and spearheaded the large consortium to understand the functional implications of the mutations in the viral genome in the context of COVID-19 disease severity. He was the coordinator of the National Consortium of SARS-CoV-2 genome surveillance in SARS-CoV and is now co-chair of the Scientific and Clinical Advisory Group to INSACOG, which tracks the emergence of new viral variants. So, dear audience, we are very honored, very privileged. So in this line, I would like again to reiterate our thanks to the High Commission of India for connecting us with such a very high caliber, top-notch scientists. So I don't have to take more time now. Leave the floor to Dr. Soumitra Das, and please, just one little information. I think this is a very real opportunity for you to ask any type of questions, especially to attendees. We have students here from the university, we have medical practitioners, we have doctors and all this. Please do not hesitate to ask your questions. Not every day that we are going to meet Dr. Soumitra Das, and do not hesitate. As much as you can, we will be here till 3:30 Mauritian time. So on to you, Dr. Soumitra Das. Switch on your mic, Dr. Das.
Namaskar. So, it's really a pleasure and honor to participate in this program. At the onset, I would like to thank the organizers, particularly the Ministry of Education, the Rajiv Gandhi Science Center, the director, and the other people who are associated with this celebration of the Indian independence, 75 years of the Indian independence, the Azadi Ka Amrut Mahotsav, and the STEM week that you have been observing for last this particular 1 week. It's real pleasure, and I'm very happy to manage time to participate in this endeavor. And I have all the best wishes for the science and technology in Mauritius. And as I was saying, that India is always happy to extend help and also learn knowledge and share knowledge with our other countries. So I would like to have a brief presentation, as is already mentioned, that we have been facing this problem of SARS-CoV-2 for last 2 years. So I will probably share my experience from 2020 till now, and how we actually handled this particular virus menace in India, and also learn from the world. I am affiliated currently, for last 23 years, as mentioned, with the Department of Microbiology and Cell Biology, Indian Institute of Science. In between 2018 and 22 January, I was heading the National Institute of Biomedical Genomics, and I actually had the opportunity to really serve the country and the world, staying as heading a Genomics Institute when the viral genomics was actually the central point to handle this particular RNA virus menace. So I would like to introduce that why this particular RNA virus, and in general, we are having stiff challenges from various types of RNA viruses. Why the RNA viruses are so difficult to handle? The major problem, what I have learned over the years as a virologist, now more than 30 years I've been working and practicing RNA viruses. So it's the high mutation rate of this RNA virus because of the lack of proofreading in the polymerase that copies the genome of the viral RNA. So in this case, the genomic RNA is copied by a polymerase called RNA-dependent RNA polymerase, which does not have the proofreading activity. If they make mistakes while copying the genome, there is no way to correct it. And that's how we ended up with more and more mutations. And that brings about the changes in the viruses. Sometimes they generate quasi-species. From one species, they make multiple species, mutants, variants. But fortunately, this error rate is at the limit of the mutation tolerability. If there are too many mutations, that particular virus cannot survive. The virus only sustains those mutants that helps them to survive better, replicate better, or infect better. So whatever is at their advantage, those mutations are selected but the others are actually, those viruses are somehow can't survive and they are selected out. So the principles of using some of the drugs you might have heard that people have been using ribavirin or remdesivir is just to increase the threshold and push it so that they can cross the tolerability level, and those mutation or those mutant virus cannot really survive and they will ultimately get eliminated. Now, this RNA virus menace is not a new thing. We have experienced long back, our ancestors, peoples have experienced in 1918 when there was a Spanish flu that was caused by an influenza A virus. And the subtypes was H1N1. There are two viral proteins called hemagglutinin and neuraminidase. And depending on their variations, they have different numbers. So H1N1, you might have heard, 1/3 of the world populations were infected with this virulent strain, and almost 50 million people we lost because of the Spanish flu. And this flu actually sustained for almost 3 successive waves, and there was a small 4th wave. And this particular pandemic again came back in 1968 and has taken lives of million persons, of which 100,000 individuals were only from America. 2009, again, we saw this H1N1 and shows up endemic settings now. And then you can, every year, almost every year, every country experiences and sees this virus coming out and killing human race. Another RNA virus that we are now listening at, just now, getting more and more knowledgeable about this particular virus is called coronavirus, which is also not very new. We had in the past SARS coronavirus. We also have Middle East Respiratory Syndrome coronavirus, or the MERS coronavirus. Actually, the SARS coronavirus was not so bad. It was 10% mortality, but for the MERS coronavirus was actually even more dangerous. It crossed about 37%. Now, the present coronavirus, which we call the SARS coronavirus 2, because the original one was SARS coronavirus 1, and this is considered as 2, was first reported in 31st of December 2019, when people were just thinking that they are getting away and getting ready to welcome 2020. And that's the time there was a report that there is a cluster having different kind of symptoms. The Chinese authorities identified a new type of coronavirus, and officially it was announced and isolated in January 7th, 2020. And that's the beginning of this whole pandemic. Now, the question is, how did we end up with this kind of a virus, which is so deadly, so difficult to handle? If you really go through the literature, there are different kind of opinion. Some opinion says that if you really go by the sequences of the viral genome and try to compare between the coronavirus that infects bat, the coronavirus that infects pangolin, and the coronavirus that are infecting us, you can see actually there are huge similarity. Pangolin coronavirus is 91.02% identical with the SARS coronavirus 2. So the question is, did we get it from the bat or we got it from the pangolin? So people found that actually there are 5 key amino acid residues, which is in one of the viral protein that helps the virus to interact with the host cell, a protein called ACE2 receptor, and helps the virus to enter the host, in the human host. And that particular amino acid mutations was actually observed in the pangolin and also in human, but not that many. And all 4 were actually conserved, not the last one, the 5th one was not conserved in the bat. Another thing that people have noticed, that there was actually additional mutation which causes a furin cleavage sites that actually helps the virus to interact with the receptor more efficiently and increase its infectivity. So that's the key words, increase in infectivity. So what we believe now, maybe perhaps it has come from bat to pangolin, and from pangolin there was a direct contact with the human, and ultimately it actually crossed through the droplets from one human to the others. And sometimes in early 2020s, we have some evidences that it were actually crossing even jumping the to cat or tiger, but fortunately that didn't really happen, or that didn't spread. But so it looks like it is acceptable, the fact that somehow we have received, or the virus got mutated from bat to pangolin, and finally it ended up infecting humans. This is again is not new. What happens, RNA virus, because of two property, which we call the antigenic shift or antigenic drift. When there is a small mutations or small number of mutations, we call antigenic drift. That keeps on happening when this RNA virus replicates. And when there is a major shift, means there is a segment of the genome is interchanged, then we call antigenic shift. Now, and another possibility is there are maybe two flu viruses infecting same individuals. They recombine and ended up with making a completely different characters of a virus. And that mutant ligand and the receptor, which becomes more deadly, it can now infect additional receptors and have a more infectivity. So a natural host, it may not be so deadly. In the intermediate host, it becomes mutated by antigenic shift or drift or recombination. And finally, it acquires the power to infect humans. And that probably happened from bat, the intermediate host pangolin, we ended up getting this virus to human. Now, so this mutant ligands infect the cells upon binding to the ancestry and accessory receptors. It's possible that it can now, it's possible that this particular mutant ligand can bind to other receptors and have more flexibility of choosing their host. So since this virus is so deadly, over the years, there are plenty of scientific literature came out. People try to decipher the virus, try to understand the genome, why this is so difficult to handle. If you look at the genome, it is actually around 30 kilobase long genome, and it has two major open reading frame, 1A and 1B. 1B is actually made by the ribosome frame shift, and there are structural proteins, which is the spike protein. All of you probably know, and I keep saying people that I learn more from the media than from the text these days, because the media educate us almost every moment. The WhatsApp University is giving you the information every time, sometimes wrong as well. But the spike protein everybody knows. And there are other three proteins called the envelope, the matrix, and the nucleocapsid proteins. Since it is a 30 kilobase positive sense RNA virus genome, the number of mutations is not expected to be very high, because usually there is a trade-off between the genome length and the mutation rate. The smaller is the genome length, larger is the frequency, mutation frequency. But even then, we are getting so difficult time with the SARS-CoV-2 virus, just because it does not have an active adaptive immune response. We don't have, our human system never saw this particular virus, so they were not ready to counter this particular virus. And coronavirus are known to shut down the interferon secretory pathway, thereby overcoming the innate immune response. We have our own artillery to save ourselves from the pathogens, but somehow coronavirus deceive them, somehow manipulate them. So our own artillery is not really able efficiently somehow protect us from this particular coronavirus pathogen. You are all familiar now with this terminology, that this virus started with the Wuhan type, and then soon there was another mutations called D614G, and that particular mutation actually caused havoc because that has more infectivity. After that, around end of 2020, we've started panicking about a virus coming out in UK called UK strain. Now it termed as an alpha. Then there was a beta strain that is originated from South Africa. Then there was a Brazilian strain originated in Brazil called gamma. And finally, another one came out from India, which actually we call it now Delta. If you really look at it, that's the virus has 4 different structural protein, as you see, spike protein, the nucleocapsid, which actually wraps up the nucleic acids, the membrane protein, and the envelope protein. Now, the spike protein is the major focus because that's the antigenicity, and also that's the protein which helps the virus to interact with the receptor, binding, and then enter into the host cells. And if there is a mutation that facilitates the virus to have a better entry, that increases the infectivity. So we talked about several mutations, we mapped them and find it out whether these mutations are actually contributing to the virus infectivity. Are they contributing for the virus to the more transmissibility or disease severity? If you really see now alpha, beta, gamma, delta, remember alpha is the UK, beta is from South Africa, gamma is from Brazil, and delta is partly contributed from India. And we see that the transmissibility has increased over the time from alpha, beta, gamma, and delta. And now even Omicron is even more transmissible. Then the disease severity was actually initially not so high, but there is an increased disease severity was noticed with the delta because of the increased risk of hospitalization. Risk of reinfection, people were already infected with Wuhan virus. Again, they got infected when the Delta virus came in for the second time. So because the neutralizing antibody that are actually generated because of the primary infection were not enough for us, for our human being to protect the virus infection by a Delta virus. And that's how the people who had Wuhan virus strain infection got it again in Delta, and now we keep hearing that there is a third time of infection with the Omicron with the same person. So there was also a threat and a problem of diagnostic or problem in detection of this mutant. If the mutation happens in the primer binding sites, as you know, all this all over the world, we are actually using reverse transcriptase based PCR called RT-PCR, where actually if you have the primer binding sites mutations, then that might miss to detect the person is infected or not. So we try to understand the impact of the mutation in the particular primer binding sites. But thinking about alpha, beta, gamma, delta, Omicron is actually super. It has actually taken out and thrown out all the other strains, alpha, beta, gamma, delta, taken out some of their characteristics mutations and have its own mutants and its innumerable mutations in the genome, and have still having the property of coronavirus with high infectivity, but fortunately low severity. We didn't really see that much of severity what we have observed with the Delta virus, we didn't really see with the Omicron. And what we do, being in a sequencing laboratory, what we do, we try to have the mutations mapped to different positions. And when they are white, that means they are not so conserved among the same strain. But when they are purple, they're 100% conserved among the strain. And as you see how Omicron is different from the alpha, beta, gamma, and delta. So Omicron has a completely different characters adopted. How Omicron ended up, or how did we get Omicron, is also a $1 million question. There are a lot of speculation. One speculation, it's possible that it might have recombined in a particular patient because of co-infection, and it might have ended up with this. And that person might have been immunocompromised, but we really still don't have a strong evidence to believe that how or to trust that this is the way that Omicron has generated, or it could be a natural contribution as well. But what we have seen from 2019 December till May 2022, you can see this is started with 19A, that's the Wuhan strain. Then it diverged into 19B and 20A. From 20A, you can see they made 20B, and there was a 21A. Now this 20 is actually 2020, and 2021 is the 21A. You can see that Delta has emerged. From Delta, we started saying them grandchildren or the sublineages are 21B kappa, 21D eta, or 21H mu. From 20C, there are beta, epsilon, iota, and 20B is the one that actually carry forward with the alpha and lambda, and then came Omicron. So if you really try to have the lineage connected, they're all started from 19A, crossed through 20A, and now we become a lineage of the 20B. And now we are having problem with 21K and 21L, which are all Omicron sublineages, which I will show you. And you can see that how worldwide this virus has evolved. You can see in the beginning, we had little bit of beta and little bit of gamma, but mostly alpha. That is May 2021. Look at over the time from May till November of 2021, you have rampant delta. But then from November 21 towards now 2022 May, you can see the Omicron has actually outcast all of them. There is very little of delta left, but all over the world is now Omicron is ruling. So the RNA virus is mutating, and the dominant mutant is actually competing out those other mutants and selecting them out. So that's how the evolution of alpha, beta, gamma, delta, and Omicron has actually survived. So this is essentially the worldwide scenario. Now I will just spend some time on what we have done. As I was mentioning that I was director of the National Institute of Biomedical Genomics in Kalyani in West Bengal near Kolkata. And that's the institute which is pioneer and championing in the field of biomedical genomics. And when I was heading that, Government of India asked us to get into this viral genomics to find out, decipher the sequence that are actually hovering around in India. So first we did a very small study with only 9 sequences from Kolkata, which is neighboring area. And we actually found and reported, and now it is heavily cited publication, that D614G and G1124 was actually found in those small population. Remember, that was only 10 sequences from West Bengal. Now, when we did that, the then secretary called me and then said, why don't you just try to have it Indian consortium to have at least 1,000 genomes sequenced in India, taking different parts of India. So what we did, we actually clubbed with 5, actually I was the coordinator, and 5 different institutes we clubbed and then tried to distribute our responsibility. And these YOLO spots are different places of eastern, western, northern, and the southern regions, and try to sequence there and find out how this virus has evolved from north to south, east to west. You can see this is the pan-India picture in 2021, where you can see the A2A, that is the D614G, which ultimately actually rampant. But we could see, interestingly, that there is a nice distribution between the east, west, north, and south. They were not identical. And that tells us that the genetic determinant, that means the host genomics, means like the host means like the individuals, differs from region to region, and that contributes whether this particular virus will be successful in establishing infection in that population or not. We also looked at the haplotypes, and you can see the north and eastern behave very similar, but west and southern India behaved differently from the north and the eastern. So there is a reason to believe that the genetic determinant of the population's distributions of India in different parts, east, west, north, south, contributed and to the severity of the disease that we have encountered for last 2 years. And that's the time around 2021 end. Government of India then decided that we should formalize this sequences endeavor. And this is for the first time, all the funding agencies, starting with the Ministry of Family and Health Welfare, Department of Biotechnology, CSIR, ICMR, all came forward, and we made this INSACOG, which is the Indian SARS-CoV-2 Genomics Consortium. And I was lucky and honored to be the coordinator, and we actually coordinated initially with 10 centers, which now extended into 55 centers all over India. We are all collecting samples, sequencing, and now we almost covered 2.5 lakhs, which is mounting around 250,000 sequences. We started with 10 sequences. Now we can proudly say India has sequenced 250,000 to actually tell the people of the world that how the virus has evolved in this huge population in India and how they cause the severity in different populations. Saying about this India, you can see that initially it was again the little bit of gamma and the other strains, but mostly it was rampant by the Delta, and now it is the Omicron. And till April and May, we can see the Omicron is actually hovering around. But the danger thing is there are now sub-lineages of Omicron started popping out. There is one that is in the media, and you might have already seen, called BA.2.12.1, and that is dangerous in terms of it's more transmissible. There are reports that it is 27% more transmissible, according to a statement from New York State Department of Health. And Omicron, there are two more varieties has come out, BA.4 and .5. Luckily, India, we don't have that many. We have one or two here and there, but we don't have it mostly found in South Africa, which is 90% genome sequenced are now BA.4 and .5. Why BA.4.5 is important? Because it has some mutations which I have highlighted, L452R or F486F, which can actually contribute to the infectivity and the antigenicity or the immune escape or the vaccine escape. Another thing that you need to keep it, remember, now worldwide we are all vaccinated. Most of us are vaccinated. But are we really protected with the emerging variants or not? So obvious question whether the BA.4 and .5 are able to be countered by the vaccinated population. But there are instances which it says that it exhibits higher resistance to the monoclonal antibody against BA.2. But again, this is Omicron variety BA.2, which is rampant in India. And we hope that since they are already exposed to this BA.2, we may not have that much of impact of the BA.4 or .5, but definitely these are all sequence-based predictions, needs to do biology to figure it out what is the consequence expected from these upcoming lineages. There are some biology, I won't get into the details, but there is Omicron subvariant BA.4 and .5 have the ability to avoid immunity induced by both previous COVID infection and vaccination, which is very, very important statement came out from South Africa. So worldwide now people are conscious to see whether .4 and .5 are being accepted, are being detected or not. But fortunately, this has not much of infectivity. It's the weakest ACE2 receptor binding activity due to this mutations in F486F and 493Q. But it exhibits stronger neutralization evasion. So that means the people who are already infected, they have the neutralizing antibody that may not be enough or strong enough to counter this BA.4 and .5. So it's important to watch out these particular mutations to prevent the another wave. It should not again put us again back into the backyard. Okay. Now this INSACOG was conceived and now it is really established and extended. We wanted to continue this function and extend the scope of this INSACOG initiative or the network. We wanted to continue as a switch surveillance because it's important when the virus becomes endemic, you need to keep on monitoring whether there is viruses in the wastewater or in the environmental samples. We are also tracking in the hospital network, and we are also trying to find out if there is any emerging mutations like I talked about BA.4 and .5, whether they are actually contributing to the immunity of the persons by the natural immunity or by the vaccine immunity. And we have built up at least 55 different institutes, and we are now, they are geared up with the facility and the expertise to sequence. So any future pandemic, we will be able to handle it and we'll be able to extend help to our neighboring countries and all the other countries if they need any help. And the knowledge management is also somehow it's very important. As I said that we are extending the network to hospital network to find out whether identify the risk factors, link the molecular epidemiology with the clinical outcome, whether this Omicron is leading to the hospitalization, their oxygen requirement, their hospital stay, whether this is more lethal to the comorbid situation. All these things are being probed at different hospitals. These are the 10 centers. Originally, we call it hub centers. And then we have some spokes at like different satellite centers, and they are now linked with the hospitals. This is a huge network. Now it's they're being coordinated by Department of Biotechnology. And we are trying to see how actually this can help to prepare Indian population and also share the knowledge with the world to protect us from the future pandemics. Now, last couple of slides, I will just show what we have done it in my laboratory at the Indian Institute of Science. You know, the virus particle have the infectious genome inside. But we have made one virus-like particle which does not have the genome inside. So it is a non-infectious, but it mimics the virus. It has all the structural protein. And you can actually incorporate the mutations that you are seeing in the spike or in the nucleocapsid, and then try to see how that particular virus would actually behave in terms of entry, behave in terms of raising immunogenicity, or behave in terms of neutralizing the vaccine efficacy. And we have recently studied that how it can causes pathogenesis. So without dealing with the infectious virus, quickly, if we have an emerging mutations, we try to incorporate into this particular platform, which we have patented, to show that we can immediately assess the impact of the mutation or the immunogenicity. Not only that, using this VLT, we have made a vaccine candidate, which we have tested in mice, and we are actually trying to see the antibody response, the T cell response, and also in the live virus neutralization. Just the paper got accepted. It will come out soon in the web, in the microbiology spectrum, which is an ASM publication, and you can look at it. But this is our very small contribution in the ocean. But what we are trying to see every time, every moment, whenever I meet the media, the question is how long this virus will take to disappear. But this is a $1 million question. How infectivity or transmissibility are being revealed by the emerging variants, disease severity, whether they are reduced or not. Flu pandemic took almost 3 waves and 1 small wave. If you go by that, we are almost end of the pandemic. But where actually there was a survey, 100 immunologists were asked by Nature on this question. Almost 90% of the responders, they believe that it will continue to circulate in pockets in world, in the global population for years to come. It may not be completely eliminated, but it may not really have an impact because the people already have either natural immunity or the vaccine-mediated immunity. So we should not really panic so much. But failure to eradicate the virus does not mean a death or illness or social isolation will continue so far on the scale seen so far. But we should actually continue to have this COVID-appropriate behavior. We have been saying it in India. We are sharing that with the neighboring countries and the other countries as well. Just we should be alert so that we don't give scope for the virus to again come back in a new form and then get them in a wrong foot. Any joint scientific ventures between the countries. So what is necessary for future, what I would think is a global network. Because now every country have developed or established a network within the country. If that can be connected, so to have a global network to help taking into account the scientific bodies of different countries, to have a continuous surveillance like SARS-CoV-2 infection. It's an example. There could be several other viruses are cropping out in different countries. And we need to screen and have a continuous surveillance and share those information with other countries so that we are not again caught into the same way that we were actually unprepared for this SARS-CoV-2. Priority will be the analysis of the SARS-CoV-2 transmission dynamics within the hotspots of the local transmission, which can accelerate the translation of the genomic data into clinical and public health. The moment we just see that there is a pocket where we can see this virus is cropping up, immediately the attention should be given to that. And by allowing a more comprehensive and accurate detection of the COVID-19 lineages from clinical and surveillance samples will help to genomic technologies and epidemiological tool and the bioinformatics tool. See, the sequence information again is an ocean, but to have a meaningful sequence information, we need to have a data integration. We need to have the patient data or the metadata so that we can correlate the sequence information to actual severity of the disease or the reason behind the severity of the disease. We can, if we have this network, we can expand our knowledge of the viruses and virus infection at the molecular and the clinical level, allow faster characterization of the emerging lineages, as I said, BA.4, .5, who knows where the numbers will end, accelerate the development of the next generation sequencing technology for future use of diagnostic assays, tracking transmission dynamics of COVID-19 in other viruses. I'm always emphasizing COVID-19 is an example. We have learned our lesson, but we shouldn't stop here. We should actually have the network in place so that we can counter any virus in future and we don't lose our friends and relatives like the way we lost in this particular wave. Lastly, I would like to thank that there is always light at the end of the dark. As you see, the illuminated NIVMG, National Institute of Biomedical Genomics, and my colleagues there who have contributed, who are actually continuing on this INSACOG activities, and my lab here at the Indian Institute of Science, who have made this VLP, or the virus-like particle, which is a wonderful platform to incorporate mutations quickly and then judge whether it would be severe or a non-severe, whether it will escape vaccines or it can able to protect it or not. So that's our little endeavor, and I'm sure with all your good wishes and all of your help, and we are also ready to help each other, we can advance the science, and that's the whole purpose of the STEM week, to increase the strength and awareness of the global community in the science and technology, engineering and mathematics. Thank you all.
Thank you very much, Dr. Das. I think this speech has been, this lecture has been very enlightening, and I'm sure it is stimulating lots of interest among all our attendees today. Like I said, we have some students here, medical students. We also have some practitioners, and these people have surely benefited a lot from what you've been sharing with us today. We have some kind of general type of questions, like people always asking, see, it's the new normal. People are still wearing masks. So, Dr. Das, what do you think? Is this going to persist? Do we still need to wear masks? Do we still need to go through restrictions? I know lots of countries are removing all these restrictions. What's your point of view on this?
Right. So I think it's important to keep an eye in the global scenario and your country scenario. If you see that it has gone into an endemic state and it is only found in particular pockets, then we actually have a close monitoring on the travel within the country, so that that pocket of infection does not really spread everywhere. So if that particular pocket experiencing, I think this COVID appropriate behavior has to be maintained in those pockets so that it does not spread. Now, having said that, we still did not really come out of this infection globally. If you really see, as on today, you can still see there are people reporting, every country is reporting some numbers, and some of the countries are reporting in large numbers. And there is no way you can continue, stop the international travel. Now, as long as the international travel is on, there will be always a fear of getting this infection from one country to other country. So I think it's the alertness. If you feel that you are facing your family members whom you are seeing for a long time and they are not infected, they are not exposed, I won't really worry too much. But if you are in a public place and you don't know who is sitting next to you, or you are receiving somebody from an airport, I think I should guard myself. Vaccine is just reducing the severity of the disease. Vaccine is not able to protect you from infection. This is a wrong thing from other vaccine. That is the difference here. That here is actually decreasing the mortality, decreasing the hospitalization, but it's not able to protect you from viral infection. I think that individual alertness is important.
So, since you've been talking about vaccines, we know that it usually takes a lot of time to develop vaccines, but in the case of COVID, the research community came with the vaccine quite fast. So, how was this vaccine developed? Can you share this with our audience today? Because it's like
Right.
people don't know much about this, the development of the
No.
vaccine.
One thing you have to remember, that this is one infection that has affected worldwide. So when any infection comes, it is usually restricted to one or two countries. And the scientists of them actually realize that there is a need to develop that vaccine. But so you can't really compare the pace or the speed we have developed a vaccine for this particular SARS coronavirus. You cannot be really compared with a flu vaccine or a pox vaccine or a polio vaccine, right? But having said that, now this particular infection has taught us the knowledge that you can actually go for a simpler technology. Earlier, people used to grow the virus, make them attenuated or defective or dead, and use that as a vaccine, right? But now a lot of times there's a challenge to grow in large amount, like hepatitis C I work. You can't really make in large amount to make a vaccine, attenuated vaccine. So people try to look for a subunit vaccine. Here for this Covishield is made up of only the spike protein. They did not really bother about the whole genome to grow and showed that it works. Moderna, they showed that the RNA, if you just make it in a laboratory, that can work as a vaccine. Those are the technologies that has been developed, is actually blessings in the boon. We are actually learning that there would be ways in future that you can make a quickly RNA vaccine to counter and make it. We can quickly clone a particular pathogenic or virulent protein and make a vaccine against that. Those are the technologies that have developed from this. But otherwise, SARS-CoV-2 is unprecedented situation. We can't really compare with the other vaccine development with the SARS-CoV-2 vaccine development.
So I think that's a very nice piece of answer for us, because as a science center, we've been also often, when we talk with students, students have often asked us how is it that vaccine have been developed. So the main point is we should not compare the development of the COVID-19 vaccine with the normal traditional way of developing vaccines. We have a question from the National Laboratory Team of Mauritius. I think it is from the Ministry of Health here. What platform do you use for the sequencing of COVID-19?
Say it again. What is that?
What platform do you use for the sequencing of the COVID-19?
Right. See, usually most of these hub laboratories are using Illumina platform. But other than that, people are using nanopore, people are using MySeq, HiSeq. There are different. So we have major centers which we call hub. They can actually sequence up to 375 or even more per day. But there are smaller institutes or smaller sequencing which they can do 25 or 50 at a time. So there are different platforms being used depending on the requirement. Now, if you are actually sequencing for the international traveler, you can't really wait to accumulate that many sequence samples and then give the results, right? So you need to use a smaller machine or a smaller platform like a MinION and try to get this information immediately to the traveler that you are actually not at risk. So, depending on the situation, people use different platform. In India, mostly Illumina based, but there are other platforms we are also using.
All right. So another question is the same from the same department. Do you screen for COVID-19 and flu for sick patients? Do you screen for COVID-19 and flu for sick patients?
You screen sick patients, or you are saying move the sick patients? What is the question?
Do you screen for COVID-19 and flu for sick patients? I mean, those who have been infected.
Flu. Yeah, right. So essentially, after Omicron came into the picture, a lot of people are reluctant to go to the clinics and get it tested. So they are thinking anyway, even if you are detected, you will have to wait for 7 days. And if you are not detected, you have to wait for 1 week. So it's not really difference between the 7 days and 1 week. But the thing is, if the person is having comorbid situation, hypertension, cardiac complications, diabetes, or any other thing which can aggravate because of this infection, even if you have a flu-like symptoms, you should get it tested. That is the advice I'm giving everyone. That's very important.
Okay. I think there's another question. I think another question is coming. Is there any steps towards metagenomics at your place? Is there any steps towards metagenomics?
Yes, metagenomics is always important, but this is more on academic interest to understand the evolution of the virus and the pathogenesis and its severity in a particular individual. There is epigenetics are also being studied. Metagenomics are being studied. Host genomics is being studied. It's very important to see that how they collectively contribute to the disease severity and link it with the variant. Because we can't really say that BA.4, BA.5 is more virulent. It may not be virulent in all the populations. It's all important on the metagenomics information that's being studied. Yes.
Dr. Das, I think the new talk of the town is about monkeypox. So regarding monkeypox, is there any kind of relationship between COVID, or do you think that monkeypox can create a mutant virus, something like this? What, I mean, attendees would like to hear from you about monkeypox.
It's very difficult to say. There are two different kind of viruses, right? There are RNA viruses, there are DNA viruses, and their hosts are different, their receptors are different. It's very difficult to say, but if you are having any virus infection, your immunity goes down. So one viral infection can make that person susceptible to another viral infection. That can't be ruled out. Like the way the COVID infected patient had black fungus. Why do you think that they got black fungus? Because their immunity was down at the time, right? So it may not necessarily one virus will bring in other virus by default or design. It could be just because of unfortunate immunity situation or innate immunity of that particular individuals, which make them more prone to other infection. That could be a bacterial infection as well.
And okay, I think we are not going to. There's probably one last. Any idea about the booster dose? Why do we need so many booster dose, ongoing booster dose?
See, again, it depends on the requirement of the individuals. As I told you that young individuals and somebody who is senior is different. Their resistance power or the innate immunity or the adaptive immunities are different. So depending on that, I think they have to take a confirmed decision on the successive booster dose. In India, it's already people are taking a precautionary dose for the people who are the frontliner. But the people who are the frontliner, the healthcare workers, we need to protect them because they are going to somehow serve us. So protect them if necessary. Flu vaccine is being given every year, right? But we don't know. It's actually, this is again a new virus. We need to watch it out for a few years to see how long the immunity lasts. So unless we see that how long the T cell memory lasts, we can't really comment on the requirement of frequent boosters. So, but definitely for the healthcare workers and the comorbid patient are in a different plate. If you have to have a decision for that, that would be different. But for the entire populations, it's definitely we need to wait and watch and see some biology, what is needed.
Okay, maybe one another question is coming up. Maybe the last one, we can take the last one here. From literature or experience, is there any functional food medicines you would suggest for us to face the COVID-19 pandemic and or any other viral pandemic? Any food medicines?
Yeah, no, that's actually very, very interesting. What I also feel that if you can just have some of the fruits which will have rich in vitamin C, if you take some of the micronutrients which are rich in some of the vegetables, so if you can do it, automatically you increase your inherent innate immunity, and that will protect you not only from virus, it will protect you for any other pathogens. Nothing wrong having a food medicines in terms of a good fruit habits, good food habits, and a good disciplined lifestyle. Majority of these innate immunity problems are coming for an erratic lifestyle disorders. So I think for the younger populations, that's the advice is you have to be little disciplined in terms of taking food in the right time and the right food in the right time.
So that's the message. Take the right food at the right time. So one last question, Dr. Das. What do you think regarding the research area around COVID, I mean, genomics? Would you advise any, if our students here would like to do some research around this area, which area would you think would be more prospective in terms of research at this moment?
Right. So if you have a scope to sequence information, you can actually try to somehow correlate with the patient information, the variants that are emerging in Mauritius, whether they are actually somehow correlated with the severity in one particular population versus another population, young population versus older population, male versus female, or even if you are going into the, a lot of times people says that people who are grown up in village background, they are much more protected than people who are actually grown up in an urban setup. So given a choice, if you have a population cohort identified in urban, peri-urban and village setup, try to see the impact of the infection in them and try to see whether they can have a correlations maintained. And we can actually learn more from them who are protected, that what food medicines they are taking, which we are not taking, and we are actually getting into a burger culture.
So that would be very interesting to know whether there is also a different lifestyle going on between these different types of upbringings. Like you just say, people from the rural areas and from the urban areas may absolutely have different lifestyles. And
Exactly.
Can that be the lifestyle may be another factor impacting on their resistance towards the COVID-19. So with that, we've been having lots of good messages from you. Today, we are very, very much privileged, very much proud to be associated to you, to be connected. And on this note, maybe Director would like just to say a few words before we leave.
Yes, I think I've been really magnetized by your discourse. It was very, especially like for me, I'm a mathematician, and it's a way for it, but we're very close together. But what I'm impressed is the way you have exposed the various the historical evolution of this genome of the genome and how it has evolved. And I think you have tried to break down the various abstract concepts in a very simple way for a non-technical person to understand. And you have broken many myths people were thinking. And for example, in some countries, they have removed wearing of masks completely. Some countries, even in Mauritius, we still have lots of sanitary protocol in force. For example, social distancing, grouping in closed areas. But I think, like you mentioned, it's just a question of protecting ourselves first. So with this, I think we look forward for more collaboration in that area. And like Hemraj mentioned, today it was a focus group, but I think we can extend it to our younger audience, because we do have lots of canvassing and sensitization to that younger level, because it is very much will take the flame for the future research and future leaders in that area of, like even in Mauritius or in India, human capital is the most important capital which you have to optimize. And this is where I think the potential lies. And thank you for your support, Dr. Das. And it was really privileged to have you. And thank you for giving your time. And thank you to all your team at IISc and your lab for doing a wonderful work for the world, not only for your community, but for the world. And we have also to thank the High Commissioner of India, who is coming up with wonderful initiatives to bring, like I said, to bring our partnerships together, increase our collaborations. Like I said in the opening ceremony of this event, collaboration is a keyword nowadays. The world is shrinking through technology, but no country can work in isolation, whether small or big. So we have to work hand in hand, share our knowledge. All the sciences now have become multidisciplinary. It's not one subject line area. And research also multidisciplinary. Even as a mathematician, I work with biologists and physicists, and everyone will put hands together for the welfare of our society. So on this note, I will thank you, and I end on that note. Thank you, Dr. Das.
Thank you. Thank you.
Okay, thank you. Just one last comment. I think one of our attendees would like to know whether through the INSACOG, would you be available to provide support to Mauritius whenever it is required? So I think that would be the Indian Institute of Science, or at any level.
Sure.
You have, within any position you have been, I don't think there will be any, we will always share your experience, your knowledge, that will be there. So on this note, a very, very big thank you to you once again, Dr. Das. It has been a real pleasure to have you here today. So thank you again, the High Commission of India, the Ministry of Education, Tertiary Education and Science and Technology, as well as all people who have been attending here today. So until another, hope to meet you again, maybe someday in Mauritius, why not physically? And so bye-bye, and
be happy to. Thank you.
everybody.
Bye.
Bye.
Have a good weekend. Bye.